خب خیلی ممنون از آقای میرزایی. Hello Bruce, welcome. کانزاس هستند که لطف کردن دعوت ما رو برای بار دوم پذیرفتند و الان ارائهشون رو خواهند داد. So take ownership and go on.
Let's see, how do I take ownership? There we go. Reza, I'm not sure I have ownership. I don't see where I'm supposed to do that.
And now you click,
there we go. I have it. Thank you.
yes.
So first, thanks, Reza and Arash, for the invitation and the organization. I'm delighted to be here and thank all of you in the audience for attending. I hope what follows is, if not informative, at least interesting. Since the 1950s, we have from the neo-Darwinian. This is a story about what selection is, how it's to be understood in terms of different fitnesses which drive because I think switching the conception of natural selection leads to better modeling practices that avoid errors in explanation, certain kinds of conceptual difficulties, and predictive failures. On the kind of account I favor, selection looks like this. I don't know that it's unique to me. I think it's intimated in both the biological and philosophical literature. But as I said, I'm for that reconceptualization for the better part of 2 decades. And today, I hope to convince you of at least one reason to prefer such a story. So I claim that fitnesses are not an appropriate stand-in for causal dependencies between rates and reproductive success, for the reason that, among other, fitnesses are measures of environmental quality and can't be reliably estimated, and even when they are known, don't reliably predict, at least for some classes of populations, medium and small populations. Let's a diploid population with selection and no mutation. So in such models, we write the next generation frequencies, P prime and Q prime, of the two alleles in terms of the current frequencies and these coefficients, the W coefficients, which are the fitnesses. So those coefficients have to represent expected per capita reproductive success for the genotypes in a particular environment if the equations are actually to successfully predict. And as such, they're measures of environmental quality. That is, those fitnesses represent something about the degree to which the specific environment conduces to the reproductive success of each genotype. And I claim as such that they lead to dubious predictions. To make that case, I'm going to use some terminology. So we will have to, in practice, we would have to estimate those fitnesses from some initial sample. And I'm going to call the period of time over which that sample is gathered the observation period. And then we use the estimated fitnesses to predict frequencies some generations later, and that intervening period I'm going to call the projection period. So with that terminology in place, if fitnesses are measures of environmental quality, what is the environment whose quality they are measuring? Well, here I am in Kansas, where the state flower is the sunflower. So let's consider a really simple population of sunflowers. We might have two types, tall sunflowers and short sunflowers, and each will find themselves in a particular microhabitat. So for simplicity, let's consider just two kinds of conditions that matter, a high and low soil moisture content and high and low soil nitrogen content. So the sunflowers in high moisture, high nitrogen content, some sunflowers in low moisture, low nitrogen content environments, and so on. So what matters then are the reproductive success here are the variables that are causally relevant to that result. And we can use that idea to define what I'll call a narrow individual environment. So a vector of values for environmental variables that characterize a particular individual. So the environment for the ith individual in our population will just be the soil moisture content value for that individual's local position and the soil nitrogen content for that individual. Once we have narrow individual environments in place, we can define narrow population environments. So those we can understand just as the frequency distribution of those two variables. So the frequency of the high moisture, high nitrogen content habitats, the frequency of the high moisture, low nitrogen content habitats, and so on. So taking a look again at our population, we have 8 organisms. 2 of them are in high moisture, high nitrogen content microhabitats, 1 in a high moisture, low nitrogen microhabitat, 3 in low moisture, high nitrogen habitats, and 2 in low moisture, low nitrogen habitats. And we can use those facts to define the narrow population environment as just the frequency of each of those pairings of values. So a quarter of the population is in the high moisture, high nitrogen microhabitat, an 1/8 in the high moisture, low nitrogen, 3/8 in the low nitrogen, low moisture, high nitrogen environment, and the final quarter in the low moisture content, low nitrogen environment. Once we have that narrow population environment in place, we can define fitnesses relative to those frequencies. So a narrow fitness is the expectation or some function of the moments of the probability distribution over per capita success for the types defined relative to that distribution of microhabitats. Unfortunately, this immediately hits 2 problems. First, the often non-homogeneous distribution of the narrow individual environments over types will vary over the projection period, so we'll be using the wrong fitnesses to predict. And second, reproductive success is a stochastic function of the environment, and as a matter of fact, we have a sample of 1. So to illustrate, again, look at our initial population of sunflowers. That population, the distribution of tall and short plants to the microhabitats will change just as a function of random assortment of the seed set to microhabitats. But even if that weren't the case, selection will change the frequencies of the various types, and so must change the distribution of the types to the microhabitats. Because the different types are differently sensitive to those environmental conditions, they are causes of reproductive success. We'll get changes in the fitnesses. So the fitnesses will change over the projection period, leading to bad predictions, even if we could estimate them. But as a matter of fact, we can't, exactly because we have in the projection period a relatively short number of generations. We can expect just one case in 5 or so generations where we actually have a particular realization of the narrow population environment. So we're estimating from a sample of one an expectation over a particular frequency density, joint frequency density for the causally relevant variables and the traits, the types, and that's just not on. So for example, if there's an association between the two environmental variables, that will matter, and we won't be able to recognize it from a sample of one. You can't calculate the correlation, for example. Okay, so clearly what we need is a story about environments on which those environments are shared widely and not going to change over the projection period. Turns out it's possible to do that. What we require is a simple shift in the set of variables we use. So instead of thinking about the particular values of soil moisture and nitrogen content, let's take averages of those values over the population as a whole. Then we have a characterization of the environment that's necessarily shared over time by all members of the population. So going back to our working example, we take the narrow population environment. We now maybe estimated from more than one generation. We calculate the average value of M and the average value of N, and then use that to produce an expectation for those averages as time goes on. So we'll call fitnesses calculated relative to that kind of environment wide fitnesses, and we might hope that this would solve our problems. In fact, that's not the case. Sadly, we have the same two problems again. First, the wide population fitnesses are unlikely to actually accurately predict what happens over to the population as it moves through a state space defined by the frequencies of the types. That's because narrow fitnesses are what's actually pushing the population at each generation. So the predictions from the wide fitnesses are going to be errant. So even if we could figure out what the wide fitnesses are, they're likely to generate bad predictions. But as a matter of fact, we can't really reliably estimate wide fitnesses from reasonable sample sizes. So that's what I'll undertake to show next. To do that, we're going to move to a different style of model. So here we want to use a stochastic process, a Markov model of what's going on. So I'm going to define a very specific stochastic process generating the changes in the population, and now to make things manageable with a smaller population. So imagine our sunflowers have a constant population size of 4, one trait variable, tall or short plant, 0 or 1, 2 environmental variables, high or low, low or high moisture and nitrogen content. And imagine that as a matter of fact, those variables are distributed as a quarter of the plants will, the probability that a given plant takes a high soil moisture content is 1/4, and the probability that any given plant takes a high nitrogen content is 0.5. To make things simple, we'll say our plants are homozygous, selfing with perfect heritability. But because the two types respond differently to the environment, we'll need different functions defining the reproductive success. I'm gonna impose these functions because they're relatively simple. Imagine that short plants reproduce seeds according to the function 1 plus the moisture content plus 2 times the nitrogen content. Tall plants reproduce seeds according to the function 1 plus 3 times the moisture content plus the nitrogen content. And then imagine we sample without replacement from the seed set to form the next generation. So we just pick 4 seeds, and that's the next generation. So that's the model. That's the generating plot process. To model it, we'll impose a transition matrix. The state of the population is given by the actual frequency of tall plants at any given time, any given generation. There are 5 such states. If the population is fixed for short plants, then it's possible at the next generation to move only to a fixture for short plants. Similarly, if the population is fixed for tall plants, it's possible at the next generation to move only to a population fixed for tall plants. But for the 3 states in the middle, and I'm not sure if my clicker can be seen, but for these three states in the middle, it's possible to reach any of the five alternatives. To complete the model, we just need the transition matrix, the probability for a transition from one state to the next for each of the various initial states. Because we have the true stochastic model to hand, we can calculate the transition matrix exactly. Here it is on the right. Those are the real probabilities of moving from one state to the other. And we can use there we go. We can use that. We can use that transition matrix to calculate wide fitnesses if we like, and move to an algebraic model. But we'll stick with the transition matrix representation. So why can't we use this information to predict the state of the population at the next generation, or 10 or 20 generations down the line? Well, let's take a look at just one case. So imagine we're dealing with a population that has just one tall plant, and we have the transition matrix representing the transition probabilities for the population in that state to each of the possible subsequent five states. That looks pretty clean. It looks like we should be able to do things straightforwardly, except that the probabilities in that transition matrix are calculated as an average over a very large number of microhabitats, microstates, narrow population environments that might realize the case of one tall plant. So there they are now on the right, all of them. There are quite a lot of them. And importantly, the probabilities associated with transitions given a particular set of values for the microhabitats for the tall and short plants will differ often quite markedly from the transition probabilities reported in the transition matrix. So it's kind of hard to see there. Let's just look at one such case. So this second to bottom row that's highlighted, this is the case where our narrow environment for the tall plant is high moisture, high nitrogen, and our short plants, two of them have a high moisture and one a high nitrogen content microhabitat. In this case, the probability that we go to fixation for short plants is about 7 in 100, so less than 1 in 10. But the transition matrix reports a probability of above 25%, so 0.28. Clearly, that's errant. That's wildly wrong. Similarly, the probability for a steady state moving from at the next generation to a population with, again, just one tall plant is just over a third, but the transition matrix reports a probability of 0.5. So those transition probabilities get it wrong, and consequently, over the short run, they're likely to generate predictions that are less precise than we could otherwise achieve. So that's a problem, but even that problem presumes that we actually know what the transition probabilities are. In this case, we can calculate them because we know what the causal mechanism is underlying the frequency changes. In the real world, we don't know those, and in fact, the standard procedure is simply, instead of to model the environment and trait causal interactions to produce reproductive success, is simply to estimate fitnesses. So we ought to care about the sampling distribution over such estimates. So imagine, again, we're trying to estimate the transition probabilities for each of these cases. What does the sampling distribution look like for those estimates? Here I've calculated the sampling distribution, estimated the sampling distribution for the move from one tall plant to one tall plant in the following way. So I imagined I had 50 generations chosen at random from those many possible microhabitats that might be realized, a sample of 50 from which an estimate of the transition probability has been calculated. And I did this 1,000 times. And the question is, how often do we get reasonably close for each of those estimates? How often do they come reasonably close to the real transition probability? So so here I took reasonably close to be 10%. within 10% of the true probability. So for this case, we get within 10% of the true probability 40% of the time. So 60% of the time, our estimate of that transition probability is more than differs from the real probability by more than 10%. Something similar happens if we try the sampling distribution for the move from one tall plant to fixation for short plants. So again, 1,000 trials, the transition probability is estimated in each trial from 50 samples. The distribution looks like this. We're within 10% of the true transition probability about a third of the time. So again, our estimates are going to be systematically be in error. All right, so to sum up, real populations, some caveats. Real populations are sometimes very, very large, and the size increases the probability that the actual narrow environments yield expectations near those of wide environments increases. And even in medium-sized populations, the reliability of predictions of long-run equilibrium frequencies aren't threatened by the arguments I've given here. That's not to say they aren't threatened, it's the arguments here don't reach to that conclusion. But many populations are not large, and often we want medium-run predictions. Furthermore, as the population size grows and the number of relevant variables increases, we increase the chance for sampling error and for under or over dispersion of the data, both of which will lead to bad predictions. So when populations are not large or samples are over or under dispersed, using measures of environmental quality, so fitnesses, offer unreliable predictions about medium range outcomes, or so I claim, and that's because the parameters those models require can't be reliably estimated, and even if estimated, do not correctly represent the causal processes driving frequency change in the population at the relevant scale. What instead we need are explicit representations of the environment and traits and the causal relation to reproductive success. And if we understand selection causally in terms of the causal relationship between traits and reproductive success, we invite better modeling practices. So I think that's it. Thank you all. Reza, I can't hear you, and I'm assuming I'm supposed to do something, but I don't know what.
Yes, sorry. My bad. Thank you, Bruce, again. And the chat box is now activated. If you have any questions from Bruce, please type it in chat box, and please write it in English.
Excellent. I've convinced everybody.
Okay. So I think no questions.
All right. Well, thank you very much. I appreciate it, Reza.
Thank you. Have a good day.
You too.
خب سخنرانی آخر ما پروفسور پیتر گاتری اسمیت هستن. اسمشون رو حتما شنیدید. یکی از برجسته ترین فیلسوفای علم و فلاسفه زیست شناسی که مقاله ها و کتاب های بسیاری رو تألیف کردن. ایشون خودشون تداخل زمانی داشت، نمیتونستن ملحق باشن، ولی برای ما فایل ضبط شده سخنرانیشون رو فرستادند که الان براتون خواهیم گذاشت.